Now we have the idea that natural plasmids, although can be used for DNA cloning. Why? Because they contain single restriction sites for different type of endonucleases. Additionally, they also contain selectable markers like the genes for antibiotic resistance. But they have certain limitations. The limitation is that the selection, it is not reliable. Although antibiotic resistance genes are there, but we discussed here that the selection, it is not reliable because many selectable markers, they are not present, maybe one or two. So for this reason, biologists realized that one can create artificial plasmids that can be used as a cloning vector. And one of the example is PBR322. So it is an in vitro constructed cloning vector, and it has been produced by using different fragments from other plasmids. For example, its tetracycline resistance gene, it is from a natural plasmid PSC101, and its ampicillin resistance, it is from RSF2124, and its origin of replication, it is from another plasmid that is having the properties of CoL-E1, and it is PMB1. So it is a chimeric molecule that contain different selectable markers and origin of replication from different sources. So its tetracycline resistance gene, it is from PSC101, and its origin of replication that mediates its independent replication from the host chromosome, it is from PMB1 plasmid. And the gene for ampicillin resistance, which is the second selectable marker, it is from another natural plasmid that is actually the derivative of CoL-E1, it is RSF2124, that provide ampicillin resistance. And these different numbers, they indicate the different base pairs that are starting are taken from different sources, and all this division is started from the site EQR1. PBR322 is an excellent example of artificially created cloning vector. So it contains several desirable properties that we are going to discuss after some time. So why it is suitable for gene cloning? तो इसकी वजह ये है कि इसमें कुछ 40 के करीब टारगेट साइट्स हैं डिफरेंट रिस्ट्रिक्शन एंडोन्यूक्लिएजेस के लिए, and mostly these enzymes, they can cut once at their specific target site. So multiple target sites are not there for a particular restriction enzyme. So if we have a look on the target sites, then we will realize that around 11 restriction sites, they are present within the gene that encode tetracycline resistance. तो इसके अलावा दो जो रिस्ट्रिक्शन साइट्स हैं वो टेट्रासाइक्लिन रेजिस्टेंस जीन के प्रमोटर में भी होती हैं. तो इस तरह से हम कह सकते हैं कि जो टेट्रासाइक्लिन रेजिस्टेंस जीन है इसमें ओवरऑल 13 के करीब रिस्ट्रिक्शन साइट्स हैं फॉर डिफरेंट टाइप ऑफ रिस्ट्रिक्शन एंडोन्यूक्लिएजेस. और इसी तरह से जो एम्पिसिलिन रेजिस्टेंस जीन है इसमें भी 6 के करीब रिस्ट्रिक्शन साइट्स हैं for different type of restriction endonucleases. Other restriction sites, they are present on other locations of the PBR322, but they are not so useful. The reason is that when we will insert foreign DNA by digesting within the selectable marker gene, suppose tetracycline, as we discussed that the insertion of foreign DNA either in tetracycline resistance gene or the ampicillin resistance gene will cause their inactivation, which is called as insertional inactivation, where the particular trait it is lost, for example tetracycline. So cell will become sensitive to tetracycline, that will act as a selectable marker, whereas the resistance of ampicillin, it will be used also as a selectable marker. So this plasmid PBr322, it contains more selectable markers as compared to the natural plasmids. So this diagram indicate different type of selectable markers again, like the gene for ampicillin resistance, and within this gene of ampicillin resistance, a restriction site for the enzyme Ph.D1 it is present. It is not the single restriction site that is present here, as I told you that the restriction sites within the ampicillin resistance gene, it may be up to 6. Similarly, within the gene of tetracycline resistance, up to 13 restriction sites may be present, and it is only indicated by 3 sites here in this picture, like sites for EQR1, BAMH1 and SOL1. So whenever we will insert foreign DNA here in this tetracycline resistance gene, it will cause its inactivation, and sensitivity to tetracycline, it will also act as selectable marker, and same for the case for ampicillin resistance if we select foreign DNA here. So this is the origin of replication that is taken from another source. The overall length of PBr322, it may be up to 4361 base pairs, and slightly it can be variable. So we can say that PBr322, it is a good cloning vector because of the presence of different selectable markers. And there is also a limitation that the length of fragment that is cloned, it is very important. So it is often mentioned that the length should not exceed 10,000 base pairs or 10 kilobase, because when the length will increase from 10 kilobase to 15,000 bases or 15 kilobase, the instability of the plasmid will increase. Instability means that it will not segregate it equally into the daughter cells during the course of cell division.